Good day, sir. I will report our Week 2 final topic entitled The Aspect of Gene Therapy. Let me define first what is gene therapy. Gene therapy is a method that may treat or cure genetic related illness. So, let's begin with a basic process. Several approaches to gene therapy includes the following. First is the replacement of mutated gene that causes diseases with a healthy copy of the gene. So, defected gene must be replaced with a healthy copy of this gene and replace the first or the mutated gene with this approach, this gene therapy. Next is the inactivation of mutated gene that is functioning improperly. So just like the first, using this gene therapy, the mutated gene or the defected gene that is not functioning well must be deactivated using this gene therapy. Third process is the introducing a new gene into the body to help fight a disease. So, a gene cannot be directly inserted into a human gene or cell. It is inserted into another gene using a carrier or a vector. A specific example was, I think, vaccines used for the COVID-19 pandemic. For example, the Johnson and Johnson are commonly known as Janssen vaccine from Belgium and US. A vaccine that is a viral vector type comes from a human adenovirus. So it used a human virus and a modified one that a vector or carrier of the gene that will be inserted through vaccines or through a vial. Let us proceed to examples of gene therapy. We have the somatic gene therapy, which involves the manipulation of genes in cells that will be helpful to the patient but not inherited to the next generation. This therapy currently focuses on recovering diseases that are restricted to the one tissue, including cystic fibrosis and adenosine deaminase, from according to Dwivedi and Pant 2018. Next, we have the germline gene therapy, which involves the genetic modification of germ cells and the origin that will pass change on to the next generation. Germline gene therapy may turn out to be most important as barrier to somatic cell gene therapy. If germline therapy were banned, researchers using somatic gene therapy might need to make difficult showing the transplanted gene could not infect the patient's germ cells and thus constitute inadvertent germline gene therapy. From International Encyclopedia of the Social and Behavioral Sciences by H.T. Greeley, 2001. Okay, what are the bioethics of gene therapy? According to Genetic Home Reference, there are four ethical issues involving gene therapy. What are these? First, how can good and bad uses of gene therapy be distinguished? Second, who decides which traits are normal? and which constitute a disability or disorder. Third, could widespread use of gene therapy make society less accepting people who are different? Fourth, will the high cost of gene therapy make it available only to the wealthy? And the last one, should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, or athletic ability? One intriguing controversy involving germline therapy is that the United States government does not fund researches on human germline gene therapy. Since germline therapy is a gene since therapy is a genetic modification of germ cells that will pass on change of the generation, and a considerable number of questions about the effects of the gene alteration of the unborn and the next generation, since the alteration can be passed on. Though experiments are done animals to animals, but no experiment or trials, the man is subject of the trial of the gene therapy. There are a lot of ethical issues in gene therapy, not just as I mentioned a while ago. Some of these are about questions on whose authority or power to decide which human trait should be altered, and other concerns are on the discriminatory effects of those who may not or cannot avail gene therapy. So, what's your view about gene therapy? That's all. Thank you. Bye.